Debra, it's great to have you here today at the Gene Editing 360 conference. Thanks for coming.
Thank you so much for having me.
It's our pleasure. I want to ask you about, in your experience, whether you've seen sort of the response from the general population to biotechnology breakthrough like CRISPR, and is it something that you anticipated when you first heard about this technology?
Yeah, it certainly has taken the field by storm. I mean, it's easier, it's less expensive, it's really sort of opened the doors to access to gene editing broadly. I do think that about 10 years ago, there was another technology that had a similar sort of floodgate effect, which was induced pluripotent stem cell research. So we had been doing embryonic stem cell research for a while at that point, but of course you need human embryos to do that work. And that is a rare and precious resource that is difficult to access. And so induced pluripotent stem cells, for which you only need a tissue sample, it can be skin, made the field much more accessible to many more people, and really sort of, as with this technology, has opened the floodgates. In terms of public excitement, I think there was similar public excitement as well.
No, I agree. It's a great example. And I know this is before your time, but in the 1970s, early 1970s, recombinant DNA became sort of part of our molecular biology culture. And it's kind of made people think about, should we be cloning genes and things like that. Is that, and I'm sure you've dug into that as we go forward, is that kind of a time that reminded you of, or now is sort of the time that reminded you of that era as well?
Yeah, to be sure. And in fact, some of the players from the early 1970s are involved in the CRISPR discussions and debates now, right? And the acylamar or recombinant DNA, this was the first time that scientists really took it on themselves to say, you know what, there are risks here. We need to think about these before going forward. And I think the scientific community is responding in a similar way with CRISPR.
In fact, I think the acylamar conference was kind of repeated for CRISPR, which was a very good sign for both of us.
With some of the same players.
Same players, right. I know this is a more challenging question, but if you step back and look at kind of the global view of human gene editing with CRISPR, are there any regulations that you think will be commonplace or that will be invoked by governments or regulatory agencies? Or are there things that we can do, or how do you sort of see that playing out?
But we're in a really different time now than we were in the 1970s. I mean, with the recombinant DNA, the policies that we developed in the U.S. really permeated the world scientific community. But the community is much larger now. And the CRISPR really, then uses of CRISPR bear on questions of human meaning. And different cultures have different understandings of what it means to be human and what is okay to do with human DNA and tissues. And so I don't think we're going to have any kind of international regulations that are able to sort of be sort of globally agreed upon, but each individual country is going to have to make decisions and engage with their public about what uses of this technology are aligned with their culture and norms.
That's a great answer, and I think we're starting that today at Christiana Care. So thanks so much for being here. Really appreciate it.
Thanks.